Hello students, welcome to Trunkie Academy. Today we are studying about the biotechnology, and with regard to that we studied about the biotechnological principles, how the different processes are involved in biotechnology, and what are the different principles and the processes that uh, has given im- has brought about a very important role for biotechnology in different fields. Okay, so today we'll study about the application of biology, and yesterday we studied about the day before yesterday we studied application in agriculture, and today we study about application of biotechnology in some other field. Okay, so other applications of biotechnology, that means the recombinant DNA technology process have made immense impact in the area of healthcare, so that means in medicine, by enabling mass production of safe and more effective therapeutic drugs. Okay, for some treatment of some diseases, okay, there are some medicines that definitely works, but for some diseases the medicines which have been discovered earlier are not 100% efficient, and with the help of the this biotechnology, some medicines or some therapeutic treatment has been discovered, and which played a very important role in healthcare sector. Okay, cloned DNA are utilized in commercial synthesis of hormones like insulin. And you know that's a very common disease, or you can say a disorder, that is the diabetes mellitus, where the human beings are not able to synthesize insulin hormone sufficiently, due to which this the blood sugar level rises. Okay, so blood sugar level rises, and this is due to the uh, less production of insulin. And what happens, this insulin is now commercially synthesized, okay, with the help of biotechnology, and that is injected into the subcutaneous region of the body to compensate the insulin that is not produced by the body. Okay, so islets of Langerhans, the beta cells are there in the pancreas, and that produce the insulin, but due to some reason, when it's not producing, okay, so blood sugar level rises. So uh, interferons are also synthesized by with the help of the biotechnology, and these are very important to fight against some kind of virus, and also to activate or to maintain improve the immunity of the human beings. Okay, so vaccines are also discovered with and have been prepared with the help of biotechnology. Okay, then these are, I told you, so insulin, interferon, vaccines are the important ones which are have played an important role in the field of healthcare sector. These are prepared with the help of biotechnology. Microorganisms are commercially exploited for the production of vitamins, antibiotics, and other commercial chemicals at lower costs. So microorganisms are commercially exploited. Mainly bacteria, you can see it's easy to handle, it's easy to research, it is do some biological experimentation in the lab, so that is the reason it is now what happens, there are groups of micro, when we say microorganisms, means the bacteria, okay, unicellular algae, and then the protozoa, the protist, fungi, unicellular fungus, and virus. So these are the microbes or microorganisms, different groups of microorganisms. Okay, so bacteria are the most common. Okay, and there are a variety of bacteria. There are some harmful and beneficial bacteria. Algae is also, some algae are unicellular, so they are under the microorganisms. Some all the protozoans are unicellular, so they are also microorganisms. And fungi, they some are the unicellular and some are multicellular, but unicellular are considered as the microorganisms. And virus is essentially microorganism. Okay, now the advantage of recombinant therapeutic do not induce unwanted immunological response, as is common in case of similar products isolated from non-human sources. So try to understand this point. Okay. Recombinant therapy do not induce unwanted immunity response, as in common similar products from non-human sources. If we take, so it will produce a kind of immunological response. Okay, it immunity will go against that, try to go against, and that treat as the foreign body. But in case of the recombinant therapeutic system, okay, this rejection from the immune system is not uh, observed, and that will be very, very advantageous for the treatment. At present, about 30 recombinant therapeutics have been approved for human use world over. Okay, so all over the world, 30 recombinant therapeutics have been approved. Okay, because there is some kind of organization that needs to approve that, because if this is in India, 12 of these are presently being marketed. Okay, so recombinant, and in India there are 12 are being marketed, and they are used for the welfare of the human beings. Now, insulin used for diabetes was earlier extracted from pancreas of slaughtered and cattle and the pigs. Okay, and that was to compensate the, I mean the, the the insulin that is not produced by the human pancreas. Human insulin is now made in bacteria, yet its structure is absolutely identical to that of the natural molecule. So what happens nowadays with the help of biotechnology, the scientists are able to, biotechnologists are able to prepare and manufacture, okay, and the insulin as same as the insulin that is produced in the human by the human pancreas. And how they have done this with the help of the bacteria. Okay, they are inserting the gene. Okay, and with the help of that, the gene when it's replicate and is expressed, they produce the insulin. Okay, so that is the way. It's a natural molecule, okay, identical to the natural molecule C peptide. Okay, then there is the alpha chain and the beta chain, and these contains 52 amino acids. Okay, insulin structure. Okay, so this is the insulin peptide chain. Okay, and the alpha chain and beta chain are bonded by the sulfide bridge, sulfide bond. Okay, so this is the structure of the insulin, which plays a very important role controlling the blood sugar level. Earlier, when the biotechnology was not much improved and not much advanced, the scientists and the biologists, the medical scientists, they used to prepare or extract the insulin from the slaughtered cattle and the pigs. Okay, now these are being prepared with the help of biotechnology and being marketed. Okay, in 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains, alpha and B chains, A and B chains of human insulin, and introduced them into the plasmid of E. coli, Escherichia coli, to produce insulin chains. So that the gene which is responsible for producing the A chain and B chain, okay, so he introduced in the plasmids of the E. coli and to produce the insulin chains, and then like you can see the DNA, then the recognizing the gene which is responsible for producing the alpha chain, then with the help of restriction enzyme cutting and taking out that particular gene and inserting into the plasmid, and then inserting into the bacterium to get the multiple copies, okay, of the A chain. Now chains A and B were produced separately, extracted and combined by creating disulfide bond to form human insulin. So A chains and B chains were produced separately, okay, so that it will be easy to understand and maintain and manage, because A chains are being produced and B chains are produced separately, so there will be no confusion and no mixing, and that is the reason. So A chain and B chain are separately, and then the A chain and B chain will join with the help of the sulfide bond, okay, and like we see the sulfide bridge in or bond in case of the A chain and B chain in natural human insulin. Okay, so the possibility of production through novel method help to have a continuous supply of insulin and stabilization of its market price. So the production novel method, this was a new method, and this continuous supply of insulin and stabilization of its market price was important. Gene therapy. Now in gene therapy, say, is corrective therapy to be taken for hereditary disease. Okay, there are so many diseases, okay, and that is hereditary disease. Okay, so that cannot be treated by taking some medicines. Okay, although some symptoms can be or some kind of relief can be given, but gene therapy involves the correcting the gene or changing the gene or modifying the gene, okay, to get the desirable character effect or to escape away the harmful effect of the genetic disorder. Okay, genes are inserted into person cells and tissues to treat that disease. Okay, so that is the gene therapy. Now gene therapy means what we are doing here, therapeutic DNA. Okay, you can see the matured DNA and therapeutic DNA, normal DNA. Okay, therapeutic DNA and the mature DNA. Now defective protein is from mature DNA, defective protein is produced. Okay, so what we are doing, we are inserting the therapeutic DNA. Okay, and normal protein is produced. After inserting the therapeutic DNA, normal protein is being produced. That is how gene therapy is done. It involves delivery of normal gene into individual or embryo to take over the function of and compensate the non-functional gene. So sometimes it involves normal delivery of normal gene into the individual or embryo to take over the function of the particular gene or compensate the non-functional gene. Okay, that's how the gene therapy works. In vivo, okay, gene therapy, direct transfer of gene. Okay, for example. So what happens? Then there is the re-implantation of cells. Okay, and and then the in vivo gene, and then from there we can isolate the patient cells lacking gene X. So what is it? We are inserting, we are in vivo gene, we are directly transferring the gene X into the patient's person. Then we are isolating the patient cells lacking gene X, and then the gene X. Okay, fraction of cells transferred to with gene X, and then the pure population of transfected, that means inserted one, is then again grown up in a population, multiplied, and then re-implanted into the cells, okay, of the human body. So that is the process of gene therapy. Molecular diagnosis. This is also very important because to know the exact cause of the disease is not easy to understand. And some diseases are such that without going at the molecular level, it's not possible to understand the actual cause of the disease. So that if the cause is known, the treatment can be planned and then devised. Okay, so molecular diagnosis is very, very important. For effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. How it pathophysiology means how it is, which is the pathogen, what is the cause, the underlying cause, okay, and how it is affecting the physiology, where it is going to harm, and how it is persisting in the physiology and affecting the physiology of the body. Okay, so ID physiology summary here you can see what is the bug. Okay, what does it do? The pathogen host is there, is it going to infect, where did it go, and how did it go, okay, and from where did it come from, and treatment. Then all these are the summary of ID pathology summary is called. Okay, so molecular diagnosis helps in understanding the actual cause of the disease, and then thereby the treatment of that particular disease. Okay, so old way, what they are doing, patient's tissue were taken, sample, okay, and pathology diagnostic. Now new way, with the help of the biotechnology, patient's tissue sample or blood sample is taken, then it's called the proteomics. Okay, proteomics, well, the proteins are observed, and then the genomics of the persons, the DNA, the genome is observed, and gene gene chip, okay, then molecular image is taken, okay, with the help of the instruments. So then we know where is the defect in case of the this genetic disorder. Conventional methods of diagnosis, serum and urine analysis, is not possible to detect disease in early stages. Okay, so if we want to know the reason of the disease in early stage, this biotechnology or molecular diagnosis is very, very important and plays an important role to understand the cause. Recombinant DNA technology, polymerase chain reaction, enzyme link, immunosorbent assay, ELISA, are some important techniques that serve the purpose of early diagnosis. Yes, PCR, okay, ELISA, and some other things. That's the important one that helps for the early diagnosis. Okay, the presence of pathogen, bacteria, virus, etc., is normally suspected only when pathogen has produced a disease symptom. Okay, so when the symptoms comes, then only, even the person who is infected with the bacteria, he is also not aware whether he has been infected with some kind of bacteria or not. When the symptom appears, when it persists, then only we come to know and we can understand, oh, this is the symptom, means this is the bacteria or this is the infected. So this is the disease symptom. By this time, the concentration of the pathogen is already very high in the body. So when the system appears, okay, till the time from the time of infection and to the time of the appearance of the first symptom. Okay, this is a sufficient time for the pathogens to increase in its population in the or in the number in the body of the host. Okay, so that is the reason we need to know early, so that we can culminate, or we can just control the that pathogens at very early stage. Okay, however, very low concentration of bacteria or virus at a time when the symptoms of disease are not yet visible can be detected by amplification of their nucleic acid through PCR. Okay, but with the help of PCR, with the help of one of the important biotechnological tool which I have already told you in the previous classes, PCR, polymerase chain reaction. Okay, so at a very low concentration also we can know about the bacteria and virus. Okay, with the help of the PCR, and can be detected at the early stage. Okay, so that is one of the advantage PCR. Okay, making several copies. Okay, PCR copies, so we can understand which is the bacteria or virus. The PCR is now routinely used to detect HIV in suspected AIDS patient. It is being used to detect HIV/AIDS mutations in the genes in suspected cancer patients. So that is the advantage of the PCR. Okay, so PCR is a powerful technique to identify many other genetic disorders. With the help of this, we are able to copy many copies, and then you can able to understand and by studying the gene. Okay, or the DNA sequence. Now, ELISA is another technique. ELISA is based on the principle of antigen-antibody interaction. Okay, so here what we are doing is antibody is the kind of molecule produced by the immune cells, or you can say white blood cell, or kind of white blood cells, and these are the actually immunoglobulin protein. Okay, 5 types of immunoglobulin proteins are formed, and they play an important role in controlling the infection and fighting against the germs or pathogens. Okay, so they have got some kind of structure like this. You can see the green in color, the Y shaped, and the upper one which has got the target antigen, that is the antigen where the binding site, antigen binding site is there. So what happened, second antibody or target, and you can see here the leveling reagents. Okay, substrate and the product. Now, in case of the sample, when it's taken, okay, it binds to its target site. Then we wash, then we label, and then we read actually. So, with the help of these techniques, we are able to, infection by a pathogen can be detected by the presence of antigen or by detecting the antibody synthesized against the pathogen. So, this is how, because by infection by pathogen can be by the presence of antigens. Okay, which type of antigen is present, okay, is being produced, we can know that, oh, that means this particular pathogen has entered. That is the reason this type of antibody is produced in the body. Okay, in the human body. So that indicates that this particular kind of infection has taken place, so that this particular kind of antibody or immunoglobulin is produced. Okay, so antibody against that pathogen. Now, DNA fingerprinting. This we studied, in the previous classes. It has very successfully helped forensic science in the search of criminals, and also solving parentage disputes. Like if somebody is claiming this is my son and somebody is claiming this is my son, so with the help of the DNA fingerprinting, it makes very easy to confirm about the parents, and also parent dispute, and also criminals. Okay, because DNA is available everywhere, in the hairs, in the skin, in the blood. Okay, and a little sample can work the magic. That means by DNA fingerprinting. So it's about like everybody has got some fingerprints is different. Okay, so similarly, like DNA, the sequence of DNA, the arrangement, the sequence also has got certain patterns. In everybody is different. Okay, although 92%, sorry, 98%, if you say, sorry, 99.9% of the DNA is same in everybody. Only 0.1% is differing in everybody. Okay, not matching. With the help of that 0.1% of DNA, the DNA fingerprinting is done. Okay, so that is it. And DNA fingerprints of some suspected criminals. Okay, we match like this. Okay, so yeah, suspect 1, suspect 2, suspect 3. So you can see the suspect 2 is the criminal, because it's matching with the crime, the DNA sequence that has been we have obtained from the crime scene, that is matching with the suspect 2. Okay, so we can conclude this. Now we come to the some important some question and answer. The number of chains that were present in insulin molecule are two chains, alpha and beta, or A chain and B chain, two. Altogether there are three peptide is the another one, but actually two chains are there. Okay, so it is the two chains. Okay, gene therapy is the correction therapy mainly for old age people disorder or hereditary disease, or for removing infectious tissue or cells, or neurological disease. No, it is a hereditary disease. There are some kind of hereditary disease, okay, or kind of some genetic disease, okay. So these are the most difficult to treat disease, hereditary and genetic disorder, okay. So this is the number. Gene therapy is used. It is used in the hereditary diseases, okay. Now, ELISA is based on the principle of antigen only or antibodies only, or antigen or antibody. No, it is based on the reaction between the antigen and antibody, because we need to know about the antigen antibody, and when the antigen is there, it is reaction, then we are able to understand this particular kind of antigen. After the reaction, then we are able to understand the symptom about the pathogen, about the germs, about the particular type of microorganisms or infectious agent, okay. So DASH is the technique used to identify the genetic disorder. Is electrophoresis, chromatography, or centrifugation. It is the technique to identify genetic disorder. Okay, so it is the genetic disorder. It is the PCR. Okay, to genetic disorder, to identify the genetic disorder, because we make the many copies of that particular DNA, and then we are able to observe. Okay, now another important application of this biotechnology is a transgenic plant. Transgenic means we are changing the genome, and we are getting a new individual, a new plant with a new genome, that is a transgenic. Okay, transgenic. So transgenic plants, we are changing the plants with desirable characters created through gene transfer. So what we are doing, we want some important desirable character to be expressed in the progeny in that particular plant, so that they will have will be used for our use, okay, like the big fruit, okay, big pulp, or disease resistant, okay, thick grains, okay, or the high nutrient content. All these characters we want in the plant. So we need to identify the particular gene which is responsible for this, or the one which is not producing. We need to identify, and then we need to change which is insert, and then this method is called and the plants so obtained are called transgenic plants. Okay, so here in this case, though a number of methods have been developed to introduce cloned genes into plant cells. Okay, so cloning, you have already I told you what is cloning. This is making a same exact copy. Okay, the number of methods have been developed to introduce cloned genes into plant cell. Okay, so T plasmid of Agrobacterium tumefaciens has been widely used as an effector vector. Vector is one that is carrying helping to carry the transferring of the these genes. Okay, so the T plasmid of Agrobacterium tumefaciens, this is widely used as effective vector for obtaining transgenic plants. Okay, several transgenic plants have been produced to meet specific needs of agriculture and human life. Okay, and this has changed of it's a new revolution, you can say, in case of the production of some important plants. Okay, so transgenic plants, tobacco is one of the example of a most common example of the transgenic plant, transgenic tobacco plant. Transgenic crop plants are having resistance to pathogens and pests, okay, which are not susceptible, and they are resistant to pathogen and pests. Okay, so papaya ring spot virus, they call the ring spot, the kind of spot on the surface of the papaya, you can see, and this is the papaya ring spot virus. Now after getting the transgenic, there has been transgenic, there is no such disease. They are resistant to papaya ring spot virus disease. Okay, so this is another example of transgenic plant, transgenic papaya. Bt cotton, I told you about a lot of things about the Bt cotton in the previous classes. Okay, how Bt cotton works, how this Bacillus thuringiensis, the name is taken from, and also the type of toxic substances, the toxic chemical substances they're produced as the crystals, okay, when it is ingested by the insects, that is dissolved in the alkaline pH of the insect, and then the toxic that is going to cause some pores in the epithelial cells of the midgut, and the insects are killed, okay, they are dead. So that is the Bt cotton resistant to, it is important one, Bt cotton resistant to insects. So Bt cotton, transgenic tomato plants are resistant to the bacterial pathogens. Pseudomonas, yeah, Pseudomonas is another type of bacteria that causes the diseases in the tomato plant, and tomato plant has also been formed, produced with the help of the gene, this biotechnology, that is transgenic tomato, okay. So transgenic potato plants are resistant to fungus, Phytophthora infestans. This is another kind of a fungus which is a very much damaging to the potato plant, and they are spreading so fast, and they are causing and damaging the potato crops. So transgenic potato has been grown now, and that is in resistant to this Phytophthora infestation fungus, and thus they are protected from the disease, okay. So that is the transgenic potato. Transgenic plants are suitable for food processing technology also, okay, because transgenic tomato are flavor savor, the bruise resistance. It is because in natural, without the transgenic, the normal on the wild type, if you see, they are easily the bruise on the surface is observed. But in case of the transgenic tomato, the surface is little smooth, it's not easily bruised, okay. Flavor savor, it is another the name of the transgenic tomato, one type of transgenic tomato, okay. So suitable for storage also, they are not easily decayed, or they are not easily they remain fresh for longer period of time, and they are easily they can also be used for a different food processing purposes, okay. So to delayed ripening and offers longer shelf life. The transgenic plants with improved nutritional value are the another successful step in the transgenic plant, because we do not only need the size of the fruits or the vegetables or the, we also need the important nutrients that is present in the food, okay, or the crops that is grown. So nutrients is also, from that point of view also, the transgenic plants are grown, and the transgenic golden rice obtained from type A is the rich in vitamin A and prevents night blindness, okay. So vitamin deficiency of vitamin A, that causes the night blindness. So this type A, which is rich in vitamin A, that prevents the night blindness, okay. So this is the transgenic rice, okay, transgenic golden rice. So this golden rice is very rich in vitamin A, and that helps in preventing the so this is how transgenic rice has been grown, okay. So transgenic plants useful for hybrid seed production also. Male style plants, Brassica napus, are produced, and this is the okay, hybrid seed production. So this male style Brassica produced with the, okay. Now, so this will eliminate the problem of manual emasculation and reduce the cost of hybrid seed. Now what happens when there are two both stamen, androecium and gynoecium there, and we want to do self-pollination, or we want to particular type of pollination, we need to cut emasculation. So when there is the male sterile brassica is formed, there is no need to do emasculation, and thus it helps in the it eliminates the problem of manual emasculation and reduce the cost of hybrid seed production, okay. We need to produce a particular kind of seed production. Once we get the particular desirable character, then we can do can produce the many copies of that. I get the next generation also, or generation after generation. We need the same type of character, so we can do that by vegetative propagation, or artificial propagation, or by asexual reproduction. Okay, so transgenic plants tolerant to abiotic stress caused by chemicals, cold, drought, salt, heat, etc. Okay, there are some plants, so they are made resistant because if there is some kind of chemicals, or the use of pesticides or chemical fertilizers, or the temperature is very low, or there is the unavailability of sufficient water, or that the concentration of salt is very high, the temperature goes rises up very high. Then what happens to all these abiotic stresses? The transgenic plants, if they are tolerant, they can grow easily, and they will not affect the economic importance, and that will be easy for the farmers to grow, and that will very very helpful. Okay, so basmati variety of rice was made resistant against biotic and abiotic stresses. Okay, so basmati variety of rice. Roundup Ready soybean is herbicide tolerant. Okay, so this is another transgenic plant which has been made resistant to herbicide. Okay, herbicide, because this herbicide, we know that there are some kinds of chemical compounds that is used for to kill or to remove some kind of herbs that is taking growing of the in the same area where this soybean plants is also growing. So they are actually being in competition with the soybean plant and taking up the nutrients. So we need to use some kind of herbicide that will destroy the herbs, that herbicide, and it will the soil that contains the nutrients will be utilized by the soybean plants fully. Okay, so this is the. And besides these, genetically modified crops have all evolved as alternative resources to industry in the form of starches, fuels, and pharmaceuticals. That also, these points also I highlighted in the previous classes. So transgenic plants have shown to express the genes of insulin, interferon, I told you before also, and the human growth hormones. Okay, so now you can see growth hormone, the okay, it acts in the growth hormone. Plants have been shown to it. So some plants, some transgenic plants, they have shown to express the genes of insulin in transgenic plants also, not only bacteria but in plants also. Okay, interferon, antibodies, yes, also. Okay, then antibiotics. Okay, antibiotic. Okay, the medicine that acts against the bacteria. Okay, bacterial pathogens or bacterial germs for the in the body. So these biochemicals produced by plants are as good as or sometimes better than those produced in bacteria. So I told you it's not only in bacteria. There's some kind of transgenic plants. They are also given responsibility, or they are these genes are inserted into the plant to produce insulin, interferon, human growth hormone, antibodies, and antibiotics. So that these biochemicals like insulin, interferon, and human growth hormone are better and sometimes as equal as they are produced with the help of the bacteria. Okay, and some are better than the bacteria also produced by the bacteria. So that is the reason nowadays the scientists and the biotechnologists are using the transgenic plants also. So utilization of plants as biofactories or bioreactors for obtaining commercially useful products, specialized medicines, chemicals, and the antibodies on a large scale is described as the molecular farming. This is another term given, the molecular. This is important for the exam. What do you mean by molecular farming? The molecular level, this is called the molecular farming. Utilization of plants as biofactories, because we are producing, we need to get a large number, a large copies. Okay, so a large number, and because we need to sell and market, to distribute in the market. So we are using it as the biofactories or bioreactors for obtaining commercially useful products, specialized medicines, chemicals, and antibodies on a large scale. Okay, in a, like industrial scale. So that is the molecular farming. Okay, so candidate protein, individual's protein, structural gene, then plant expression vector. Okay, now introduction. A vector will be used to introduce in the plant, and then the transgenic plant will produce will be planted, the harvest, and then we then we extract and purify that from that particular plant, and then we prepare the pharmaceutical as a medicine. Okay, so it is used. Now in the near future, this field is expected to revolutionize both the farming as well as the biochemical industry. Definitely, it looks like that also. Now we will go through some questions. Plants with desirable characters created through gene transfer method are called transgenic plant. Okay, it is called a transgenic. Okay, it is a transgenic plant. So it is very easy question. Now the vector which is widely used to produce transgenic plants, it is PBR322 or col E1 plasmid or bacteriophage or T plasmid. So the widely used is the T plasmid. Okay, is a T plasmid. Okay, with the help of this only transgenic tobacco was obtained. Okay, so it's a tea plasmid, widely used factor. Utilization of plants as biofactories or bioreactors for obtaining commercially useful products is described as biofarming or molecular farming. It's a molecular farming. Just now we studied about this, isn't tissue culture and biotechnology. It's the molecular farming. Okay, so this was it. So now this was all about the applications of biotechnology in some other field apart from the agriculture. So we studied about that application of bioculture finds its application in the field of medicine, like the production of insulin, okay, transgenic plants, okay, and some kind of therapeutic medicine or therapeutic use, like when we find some kind of genetic disorder, we want to change that particular type, or we want to in the embryo or in the adult stage also. So there are different types of use of biotechnology, and this is has changed, completely changed I mean in the medical world, or you can say the treatment, the way of treating the different diseases. Okay, the diagnosing the different diseases. Okay, and also the producing the crops. Okay, and also fighting to control the effect of the pests and the fertilizers. Okay, so all these things, it has been very much beneficial and effective in these fields, as I told you, in agriculture, in medicines. Okay, and so in near future, okay, if the way the biotechnology is getting advanced, okay, so we should hope that there will be some more new surprising things to come, and important way of and controlling the different harmful diseases, and also the protection of the plants from different types of diseases, and getting a very good and extra good quality of the crops. That is the demand of the need of the hour, because the population is increasing and the demand for the crop is also increasing. Okay, so this was for the today. And now in the next class, tomorrow again we will continue with the next topic. Okay, so tomorrow we will meet again at the same time. Okay, and now you can till then you go through this DPP and answer the question and send back to us. Now, thank you for joining and listening. Tomorrow again we'll meet. Till then, goodbye.